Questions for Dr. Shilsky?
Rich Shilsky from ASCO. Over here, David. So, we've heard a lot of discussion today about testing, regulating testing, qualifying testing, and so on and so forth. Most doctors and patients don't have a clue about what test is performed that's guiding the treatment decision that the doctor makes with the patient. And I guess the question that I have is, if I were a patient, how would I know if I've had a quality test performed on my tumor? What information should I ask? Who should I ask it of? And what response to my question would tell me if a quality test has been performed?
Okay. Thank you for the question. I think that's it's a tough one. I mean, right now, patients are left to ask their physicians or whoever else is involved in testing or their care about their tests. But as you mentioned, I think even physicians are not aware. I mean, I actually, every time I go to the doctor now, I sort of ask about the testing, and then I tell them I'm from FDA and describe LDTs and stuff, and they're like, wow, I had no idea there was a difference. Yeah, exactly. Sometimes I'll get very long lectures, and I didn't know they had the time to spend with me. So, because but I do think, I mean, I think your point is well taken. I think right now there are little disclaimers on an LDT test report, but by the time that gets through the medical record, it's not there. I mean, we would hope physicians and patients will start asking, and those questions will go up the chain, but I do think we need to work out how this communication is going forward, not just about the analytical validity, if it's a good test or not, but what does the evidence say? And it's hard. I mean, even coming up with a uniform statement about what a good test, I think, is challenging, because that depends on the disease and all sorts of factors that we can't predefine. I mean, hopefully people will begin to understand there are sources of information. FDA publicly posts decision summaries, that they'll understand the difference between a test that's been FDA approved and hasn't been FDA approved, and we can go from there.
The simple answer will be that ASCO needs to educate all the oncologists. Okay, we can talk about it.
I think it was over there first, yeah.
As an economist, there's something really interesting going on here. Catherine Phillips, University of California, San Francisco. On the one hand, we have one company that's really making all of the machines. That's Illumina. They clearly dominate the machine space. Not only that, you've cleared their machine, which is a huge barrier to entry for any other company. But on the other hand, we have all these different clinics, companies developing all these different tests, all different ways. So we have a very narrow market on one hand, but a very wide open market on the other hand. So how are we going to resolve the difference between the two?
Well, I think, I mean, I've always thought of them as two different things. I mean, I think Illumina runs its own LDT right now. And I forgot to mention Ion Torrent also now is cleared as well. So we do have two instruments going on, but I do sort of see a world where you will have components manufacturers. I mean, this is what I predict, and I'm never wrong. But we'll have the components manufacturers, but those still need to be assembled into tests. And so you will have labs that will be, again, choosing from these components. I think it's going to, putting together kits is one way. They could put together kits like other tests, or you'll have labs purchasing from components. But I don't see the labs as really competing with companies that make components. They need those companies. But I think there's like separate things.
Some people believe, though, they're going to merge into one. Illumina is developing its own tests now.
Right, they offer that as an LDT
Right.
now,
Yeah.
but I mean, that certainly hasn't at least yet supplanted. And I think one of the issues is the technology is moving so quickly, in my mind. I don't know if you can freeze it and say Illumina's going to have the monopoly on all this. And, there still continues, I think, to be disruption. And we see some things coming down the road like long reads and things like that. And I think there's still a lot of new players coming up. So I think it remains to be seen. But I do think there will be a role for components manufacturers independently of the labs, that they're a necessary part of this.
So my take on the current, landscape is that we spend extraordinary efforts to develop companion diagnostics, but no one ever uses the companion diagnostic. And I assume that the FDA recognizes the fact. So that seemed to me what this session should have been about, and it wasn't mentioned. So I hate to put the FDA guy on the spot for the next question, which is, what is the FDA's position on the fact that no physicians, in fact, order the companion diagnostic for the drugs that are already approved? And just a quick example for those in the audience who don't know that, nobody orders the BRAF companion diagnostic before they put their patients on the BRAF inhibitor. So again, that seems to be very obvious in the marketplace. And is there a regulatory view on this? Because there seems to be a standard guidance that doesn't seem to fit the reality in the clinic.
Well, I think in the first place, we don't, that is essentially, you could think of that as off-label use, which we don't regulate. Physicians are free to order whatever they want. So at that level, that's just not something FDA gets involved in. Certainly we're aware that this happens, and that, we are just sort of from the standpoint of not knowing which tests are good and which aren't, that is, I think, of general concern, not just to FDA. But, I think in the current environment, there's, as long as there's enforcement discretion on LDTs, companies are free to develop them, and physicians are free to use them. There just isn't I think, awareness that the technologies aren't the same, the results may not be comparable, and so you can't guarantee that the result you get from any one of those other tests will be predictive of the same response you saw in the trial with the approved test and the drug. So I think a lot of that is also, again, education. But as an agency, we only review what comes in, and we don't regulate practice of medicine. So physicians are certainly free to use it.
So just to throw out a follow-up comment, it would be my recommendation to the standard setting people who are in this room, people who are heavily invested in NCCN or maybe ASCO or ACR, that they formally make a recommendation that the current COVAS companion diagnostic is actually a very poor test and shouldn't be used. And I'd love to see those groups go on record to say that the actual companion diagnostic is inadequate. It's a poor test, doesn't actually pick up even all the V600E alleles, so that we could really bring clarity to a situation in the clinic, which is even more confusing for clinicians, where you have a regulator suggesting a test that the experts in the field feel is completely inadequate.
Yeah. Well, I think it would be helpful to know, and I think this is one of the issues, is there just hasn't been a lot of transparency with LDTs. So I think from practice what's good and what's not. The clearance of the COBAS test was done because that's the test that was used in the trial, and it's reported with all, it's not, no test is perfect, and I don't think we would claim that. We're not trying to say that LDTs are necessarily worse, the other LDTs out there. It's just we don't know, and that's sort of our position. So I think if people know that, at least for the time being, they should share that data and make those recommendations, and that would be very helpful.
If I can add a little more, I mean, these drugs that were approved based on companion diagnostics, I mean, when you have a companion diagnostics, that basically narrow what kind of population are you treating. Doesn't mean that there may be some other mutants that could be predictive for the antitumor effect. But when you have a companion diagnostic that you identify that population, you know what kind of response or antitumor activity you have with this novel agent in that particular population. And that's what it's all about. Certainly with the new understanding of the targets and the drugs, there is some evolution, as I mentioned to you, with the HER2, and that there was an initial test that it was insufficient, and science have evolved. And I think this, and as part of the organizing committee, we wanted to bring this to the attention to the folks about this LDT, because again, it's I won't call it a fight, but there is different stakeholders have different mindsets. These companies that created these companion diagnostics, they realize that physicians are not using this, and certainly they are, I don't know if they're losing money or not, but they are unhappy. However, academicians who have created some of these LDTs don't want the FDA to regulate them because of, and there is a significant, and it will be hopefully compromise. And I think the compromise, and if I understand what the agency wants, is that for LDTs that are commercially available tests, there's going to be some kind of regulation over time. And I think it's a reasonable compromise. And for example, for the COVAS or whatever test you think, if you're going to come out with an LDT, a new LDT, certainly it is a better test, supposedly. You will need to have some clinical validation, clinical data, and if that's the case, that's what's going to be used. I don't know. That's my interpretation, but I don't know if I'm right or not.
So this is a great question, because I think it brings up a larger concept as well, which is that the fitness for use, and that a companion diagnostic, part of the fitness for use is to get approval. And so the characteristics may not be exactly the same as what would be optimal fitness for use in a clinical testing situation ultimately. And so actually Mickey Williams earlier in his talk mentioned that for the MATCH trial, they were looking for maximal specificity, because that was part of what they wanted to achieve in the trial, and sensitivity wasn't necessarily the main focus, and they had ways of dealing with lower sensitivity issues. If that same test were brought into the clinic, sensitivity, if we'll use lung cancer again as an example, if you're dealing with very tiny biopsies or very low tumor content samples a lot of times, then sensitivity may be a huge issue. So some of this may come back to kind of what the development path and the needs were to be accomplished during that time as to why a particular test got approval.
Dr. Mendelson.
John Mendelson from MD Anderson. I have a question for Dr. Litwack. Dr. Eberhard presented his friend, and there were some gene abnormalities that could have led to off-label use of drugs. And that's an N of 1 situation that's up to the physician and the patient. And if the drug's approved, if he'll pay for it, and if it's not approved, if the company will release it, which is very unlikely for salivary tumors. It'll be hard to get the drug. But there are many people now designing clinical trials where they're trying to, we have a huge amount of data available now. We can get DNA data, we can get RNA data, we can get expression data, we can get protein, phosphorylated proteins, reverse phase array data. And we're all frustrated about the fact that half the time with standard chemo, much less with the targeted drugs, you don't get responses, even though the other patients do respond. So we're designing trials where we're trying to collect data and assign patients based on expression data or reverse phase data. And my question is, does that kind of a trial need to go to your branch of the FDA? Does it need to get device diagnostic permission? As long as the tests are being done in a CLIA environment and they're reproducible, do we have to go to the FDA not only to get clearance to do the trial, but do we have to get clearance to use the tests for that trial?
Well, I can say definitively it depends. The
I think it's a changing approach.
I think we've, as we've gotten more experience with companion diagnostics, I'd say we refined it. So the regulations are now not about marketing, which is, I think very confusing with LDTs and FDA-approved tests. This is now about whether a test is investigational.
Right.
And is it being used in a clinical protocol? And if those are the cases, then we look at the risk of the test. So just because you're using really new technology for serious illness doesn't actually mean you would necessarily need an IDE, as Adrian very nicely described. If we determined, or you or your IRB determined it was a significant risk study, then we would require an IDE submission. But it's really, in that case, we're really looking more at the risk that's posed to patients from how the test is used, not what the identity of the test in often cases doesn't even matter. It's like, are you using it in a way, like in an oncology trial, it would be, are you making a clinical decision or assigning patients?
Well, sure, we are making.
Yeah.
We're picking therapy based on a pathway that's been identified, relevant for that drug, but there's no proof
of course,
that test correlates with response to the drug.
Because you wouldn't be doing the investigation
right,
otherwise.
exactly.
Yeah.
I mean, that's the conundrum we're in.
But I think that there are a number, in fact, we're hoping to release a guidance on this in the next few months, and we're talking about this a lot. So when we're looking at risk, we're not just looking at, are you sending somebody to treatment, but like, is this a patient who has exhausted all of the treatment options? Their risk is much less because there are no other options. The risk of the false test result is much different than if you were, say, looking at this in first line, as a first line treatment for breast cancer, and you were using the test to determine that would be a much different situation. Some drugs are highly toxic, others are less toxic. That's going to go into risk. So we actually look at now at a number of factors not just whether it's a selection trial or not in determining significant risk. So it is, there's a lot more that goes into that now.
Right. So what you're saying is you're balancing risks and benefits, which is music to my ears. If a patient has advanced cancer and has failed standard of care,
right,
the benefit outweighs the risk compared to somebody that's de novo with cancer.
Well, actually, it's a complicated distinction. We actually are only looking at risk because there are certain things like the number of patients. What we're saying at this stage is, do we need to do a full risk-benefit analysis?
Right.
If it's non-significant risk, we say, no, the FDA is out of it. Just do a few things and you're fine. If it is significant risk, we say we need to review this and make sure the benefit risk is appropriate. So early on, it makes it a little different, which would be a much longer discussion than I think we have time for.
So your advice now is submit it and get a ruling of whether we need an IDE?
Do a pre-sub.
Because there's hundreds of trials like this being planned.
I know.
Do you want to review that for IDE requirement then? Is that your advice?
Yeah. Well, I think right now what we're doing is we're talking to IRBs so that they understand what it is. I mean, the way we actually look at risk, we shouldn't be seeing most of these trials. Most trials are either exempt completely or are non-significant risk. The problem is there's been a lot of uncertainty. So what we want to do with the guidance, and I personally am like talking to IRBs and giving seminars, webinars on this is, this is how we look at it. Because this is where the decision actually is supposed to happen, not at the FDA level. So our goal is to get everybody to the point where we don't have to make
Good. Well, I hope the
decisions.
guidance comes out soon.
Yeah, I do too.
It's a huge barrier every time you think about these kinds of
things.
things.
I agree, and we want to get it out. Right.
Dane Dixon from the Palmetto MolDX program. What is the role, do you think, of an entire medical community in trying to educate physicians and patients that certain testing may be harmful to them, particularly when companies that do not apply, and most companies that are marketing tests to physicians that were originally meant to be used in-house as a laboratory-developed test, and they don't go through and get any regulatory approval, and then they come to physician's office with marketing materials, and the physicians are assuming, in many cases, particularly in the community centers, that the data that is being presented to them by this laboratory is the equivalent to what has been presented in a new drug application to the Food and Drug Administration. And so there seems to be where we've got a huge disconnect, where we have laboratory companies that don't want regulation, partly because they have the ability to market whatever they want based on whatever data they have, even if the data, when you drill down to it, is reasonably thin. There's also a reasonable unblinded trust in pathology by many medical oncologists who say, well, I don't know what the methodology is. I don't know if it's LDT or companion diagnostic or whatever it is. Whatever they gave me, that's right. What is our role to help to protect the patient and protect the science in such a way that we help educate that perhaps pathology and what we've been doing in pathology for so many years is not entirely right?
So I guess off the top of my head, it seems like there may be two levels to your question, and one level is the regulatory level. So it's your turn now. No, the other level is at the level of oversight of testing laboratories themselves. And for better or for worse, this has really been taken care of by pathologist organizations themselves. The CAP has its laboratory review and accreditation program, et cetera. That's what the CMS CLIA regulations are about also, as far as how to establish a quality testing environment that does things in such a way that it doesn't do tests that should be or that could be harmful to patients. So that's the pathologist laboratory level. Should there be another way of policing individual laboratories? Is the professional organizations, are they not doing a sufficient job of providing quality testing? I mean, that's certainly something that we need to constantly ask ourselves and to make improvements whenever they're needed.
Well, because it seems to me, I mean, we're seeing what I'm going to call a dichotomy. We've got pathology groups that say we're fine, we're fine, we're fine, we're fine, we're fine. We don't need any more regulations. And then we have the FDA who comes out and says we're concerned about the quality of the testing, not because you're not doing it right, but because we just don't know. And then we have a lot of these major trials like the NCI, and many of these trials are saying we are going to develop standards and very strict standards because we need to know what we're testing for. And so when we're looking and we're really drilling down to it, we're saying, okay, well, that's different. That's a clinical trial. And yet I'm looking for a clinical trial for mutations. And we know from previous experiences that, that most tests, and particularly molecular tests, physicians don't know what to do with anyway, even inside of academic centers. There's some recent data that says we don't even know what to do with this information. And so I guess the question is, what is our job to go through and say, who is the right person to do this? Is it CAP? Is it ASCO? Is it NCI to say, maybe we need to hold someone's feet to the fire if we feel it is not the safest way to do it? And if it is, then great, let's move forward.
Just one more comment on that is a trend that does have pathologists concerned are clinicians bringing testing labs into their own practices and performing tests there, which is another level, and that even concerns pathologists.
So last question.
Hi, Federico Monzon from Invite. I think I had another question in mind, but I'll just make a quick comment about what was just said, because I think probably most people here don't realize how many LDTs are out there, and the large majority of oncology tests, the large majority of genetic tests are actually performed on LDTs. There's, millions of patients that are benefited by LDTs being performed responsibly. And maybe we're talking about, a few cases in which, yes, there might be laboratories that are, pushing some of their tests that might not be, clinically validated. But I do want to paint the picture that the way patients are managed right now is safe, and a lot because of what Dr. Eberhard said about the regulatory environment and the own pathology organizations which regulate our or which allow us to monitor and policy ourselves on that regard. Now, so the question I was going to ask is, you made, Dr. Lidback made an important show of how the technology moves and how these changes, and probably, technology moves faster than what our regulatory environment can do. And, I think it was already pointed out about the BRAF issue. This goes to KRAS. This goes to all of the biomarkers that we're using now, right? Which some of them have FDA approved assays. But what happens now when we move in these multi-analyte testing, right? So there is a predicate device for KRAS testing. There is a predicate device for BRAF testing. But now I'm doing a multi-gene panel that includes PI3 kinase, KRAS, BRAF, all of those things. And, how can I move forward? Because if there is if I do a PI3 kinase, currently there's no predicate, right? So I would move into a different category of things. But if I do all of them together, what happens? What is the pathway I'm going to follow?
Well, I mean, just traditionally for multiplex, multi-analyte tests, we look at each analyte separately. Now, at some point with the gene panel, you're going, I mean, whole exome is a good example. That's just a really very large gene panel, but we couldn't expect you to provide analytical data for every one. So at some point it becomes a discussion with FDA, which is why we have these processes available to have this discussion about, well, what is the data you can get and what do you need to show? I mean, we might move to a representative approach at some point, but I think it is going to depend again on a number of factors and exactly how the test looks like. But we haven't cleared or approved a cancer panel yet, so I would say that will be the model. I mean, I would agree with you that the single tests that you're talking about, their requirements are going to look different than the large panels. And I assume you're thinking about, cancer panels, gene panels determined by NGS. Yeah, I think that is something we can handle from a regulatory standpoint. But I think, once, it's oftentimes it awaits the actual submission to finalize how that is going to look. But I mean, at some point we would transition, I think, from every analyte to representation. So that would be my answer right now.
Okay, we concluded the most important session again today, and we're coming back at 1 p.m. So we have lunch outside. Thank you.